Good morning, guys. Now we are going to solve some problems on the monohybrid cross. Just to remind you, with the monohybrid cross is the inheritance or the crossing of individuals that are different only in one characteristic. This characteristic is controlled only by one gene which occupy one locus on the chromosome. We have here an example. In the pea plants, spherical seeds are dominant to the dentate seeds in a genetic cross of two plants that are heterozygous. Both plants were heterozygous for this characteristic for the seed-shaped trait. What fraction of the offspring should have spherical seeds? Okay, so I have spherical seeds with spherical seeds. Both of them were heterozygous. You know, the alleles will segregate in gametes. So this will be the distribution of alleles in gametes. And here is the result of the crossing. You can see that the ratio here, the genotype will be capital S, capital S, capital S, small s, and small s, small s. This ratio, the ratio of the phenotype is 1 to 2 to 1. While the phenotype ratio, if you have the capital letter or the dominant allele, so the dominant characteristic will be inherited, the other one will be hidden. So I have here 3 spherical, 2, 1 dentate. Okay, the question is asking, what fraction of the offspring should have spherical seeds? So how many should have spherical seed is 3/4 of the offspring. What is test cross? Test cross is when you make breeding or crossing between an individual carrying the dominant characteristic, but we don't know its zygosity. Is it heterozygous or homozygous? We are going to cross this individual with another one which is recessive homozygous. Of course, all recessive are homozygous individuals. And then we are going to analyze the proportion of individuals in the offspring. And based on this, you will predict if the individual carrying the dominant characteristic was heterozygous or homozygous. So suppose we have red flower plants. Okay. And you know that the allele for red is dominating the allele for white. So in order to know the zygosity of the red flower plant, you are going to cross the red flower plant with the recessive homozygous, which is white. And then look for the offsprings produced in the first generation. Analyze the proportion of red or white. Based on that, you will be able to know the genotype of the red flower plant. Draw the Punnett Square and draw your alleles. Distribute the alleles in gametes. Here are capital and are small. Okay, like this. And then look for the genotype of the offsprings. We'll do the same thing if the parent was homozygous. If homozygous, all gametes will be carrying the same type of alleles. So 100% here, 100% of the F1 generation will be red. While here, you will have 50% of the offspring were carrying the red, while the other 50% were carrying the white color. This means that based on the proportion of the offspring of the phenotype of offspring that you will get in the first generation, you will be able to decide the zygosity of the offspring. If all the generation were carrying the dominant characteristic, this means that the parent should be homozygous. While if you get the dominant and recessive characteristic in a ratio 50%, 50% or 1:1, this means that the parent, the one who was carrying the dominant characteristic, was heterozygous. Okay, heterozygous, and here if it's homozygous. Let's take an example now. To identify the genotype of yellow seed pea plant as either homozygous dominant or heterozygous, you could do a test cross with plant of genotype. Which genotype? These are the possible genotype of the unknown parent. So in order to know the zygosity of it, so which genotype you should be crossed with? Yes, it should be crossed with the recessive one, which is y small, y small. We have one more problem here. Monohybrid cross problem, a test cross used to determine if the genotype of a plant with dominant phenotype is homozygous or heterozygous, if the unknown is homozygous. If the unknown was homozygous, all the offspring of the test cross have, yes, it should have the dominant phenotype. If the unknown heterozygous was heterozygous, so half of the offspring will have, if it's heterozygous, 50% will have the recessive characteristic and 50% will have the dominant one. So I have here recessive, so E is the correct answer. Now let's solve some of those problems. First one, in angelfish, the red is dominant and green is recessive. A homozygous dominant male is crossed with a heterozygous female. What is the ratio of the genotype in the offspring? We are going to use this square to solve all of those questions because this is the square that we use for all monohybrid cross inheritance. Genotype of parents, he said homozygous dominant. Okay, and the red is dominant. So I'm going to use capital R for the red color, and as he is homozygous, so capital R, capital R. While the other parent, he said heterozygous female. Heterozygous female, so capital R, small r. These are the genotype of parents. Phenotypes of parent, they will be red, both of them. Okay, this was the male and this was the female. Okay, in Punnett Square, you are going to show all gametes, all probabilities of gametes. For the male side, you have capital R, and for the female side, I have capital R and small r. Make the cross, put all the probabilities, and then write the genotype ratio. You can see that I have capital R, and capital R, small r in a ratio of 2:2 or 1:1. While the phenotype ratio, all of them are 100% red. Question 2. In squirrels, gray is dominant and brown is recessive. Okay, so as a gray is dominant, so I am going to use capital G for the gray and small g for brown. He said homozygous recessive female. So the female was homozygous recessive, so small g, has been crossed with heterozygous male, so capital G, small g. This will be the genotypes of the parent. How many of their offsprings will be brown? Okay, let's complete our diagram. The phenotype of parents, but here it's gray and the female was brown. Okay, draw all the probabilities in the square of gametes. So I have here capital G, small g for the male, and for the female we have only small g. Put all probabilities and fill the square. Okay, so the genotype ratio, as you can see here, I have capital G, small g and small g in the same ratio, 2 to 2 or 1 to 1. Phenotype ratio, okay, also we have gray and brown, okay, 2 to 2 or 1 to 1, so it's 50% to 50%. So he's saying how many of the offsprings will be brown. Of course, we don't have the number, so we are going to use the ratio or the percentage because you didn't get the number here. But there will be half of the offsprings will be brown. Then a pair of anglefish produces 400 offsprings in one meeting. 300 of them are red and 100 are green. What does this mean to you? If I have 300 red and 100 green, this means that the red is dominating the green. So we can use capital R for the red and the small r for allele for the green. First question, what is the genotype and phenotypes of the parent? Okay, he said there were 400 offsprings, one pair of anglefish. 300 of them were red and 100 was green. This ratio is 3 to 1 ratio. You get only 3 to 1 ratio if both parents were heterozygous. So I expect that both parents were heterozygous. And what about their phenotypes? Both of them were red. Okay, and then prove it with the square. Yeah, we need to prove this ratio with the square. Okay, so put your gametes in circles and match. You can see here that we have 3 red, the phenotype ratio, 3 red and 1 green. So we proved the phenotypic ratio with the square. Let's solve now question 4. Another pair of anglefish produces 400 offsprings. 198 of them are green and 202 of them were red. What is the ratio of the phenotype? You can see that the ratio of the phenotype is almost 1 to 1. Okay, because we have almost 200 green and 200 red. So question 1, the ratio, so I will write 1 to 1. What is the genotype and phenotype of the parents? He said the phenotype is already given. You have green and red. So this will be the phenotype. So the ratio of the phenotype in the parents will be red. So I will write here the phenotype of the parents, red and green. What will be the genotype of the parents? Look for the ratio 1 to 1 that you get at the phenotypic ratio. This phenotype ratio you get it only if you have one of the parent is homozygous and the other one is heterozygous. So of course, as the red is the dominant, so the red is the one which might be heterozygous and the green is the homozygous one. Okay, these are the genotypes and phenotypes of parents. Prove it with the square. So we need to prove by the square that you will get a ratio 1 to 1. Okay, so fill the squares as normal, put the gametes in circles, R capital, R small, and okay. So you can see here that I have the phenotypic ratio. I have 2 red and 2 green, means 1 red and 1 green. Now let's go to problem number 5. Assume that a certain type of squirrel could have up to 80 offspring in one batch. If 21 of 80 babies are brown, that what is the ratio of the phenotype of the offspring? Predict the genotype and phenotype of the parents and show your work. If this batch contains 21 of 80 babies are brown, this means that I have 59 babies or the other feature, which could be the black one. So what is the ratio of the phenotype of the offspring? Okay, you can see here the ratio. You can make it approximately to 20 to 60. So to make it in the sample ratio, it will be 1 to 3 or 3 to 1, because the brown is a recessive and the black is the dominant here. Most of the individuals were black. So this will be the ratio of the phenotype in the offspring. Predict the genotype and phenotypes of the parents. If you got a ratio 3 to 1, this means that the parents were both heterozygous. So if I use the capital B for the dominant one, so both parents were capital B and small b, because this is the only cross that can give you a ratio 3 dominant to 1 recessive. Show your working. You are going to draw the Punnett square as normal. Show all the alleles, distribute all the alleles in gametes, show the ratio of the phenotype and genotype. Then let's go to question 6. You buy red belt newt from a local pet store. You know that the red one is dominant, okay, and the yellow is a recessive. If you don't know what are the parents of your red one, what can you do to determine the genotype of your pet? Okay, so you got the one which is dominant, but you don't know is it dominant heterozygous or homozygous. You are going to cross. You make test a cross. Test a cross means you are going to cross your red pet with the yellow one, which is the recessive. Okay, so red will be crossed with yellow. This will be the parents' phenotype. Okay, so for the parents' phenotype, red will be crossed with yellow. Genotype of the parent, here we have 2 options. The red might be capital R with the yellow, which is small r, or can be heterozygous. You should make both trials. Okay, so here you will draw then your square showing the distribution of alleles in gametes. So if the parent was homozygous, so you expect to get 100% of your offsprings are red. This means that you are going to cross your pet with a yellow one and look for their offsprings. If you got a ratio 100%, this means that your pet was homozygous. But if you get it 50% red and 50% yellow, so we expect it to be heterozygous. Okay, so here the parents, you know, the genotype and phenotype, everything was similar. The square. So the result of this cross will be red to yellow in a ratio of 1:1. So in this case, so you can predict the genotype of your parent by making the test cross. Inshallah, we'll continue the lesson in the next video by explaining the codominance and the ABO blood group in human. Thank you.